The social context in which mate choice occurs can influence reproductive outcomes and may do so in different ways for males and for females. In species with lek mating systems, males frequently perform courtship displays when multiple females are present. For example, on the right, you see a male Corolla's parodia, who has a literal line of females watching him display. Leks are mating systems where males mate polygynously and take no part in parental care, but perform displays in reliable locations. Some males on leks are far more successful than others, so leks are models for the study of sexual selection. Though lekking species of many types have these multi-female displays, it's been difficult to figure out why females sometimes attend displays together and how it affects prospects for males. The presence of other females could provide information about preferred mates, creating the possibility of mate choice copying, a phenomenon that was actually first suggested in the greater sage grouse. The presence of multiple females could also result from competition for preferred mates, as in topee antelope, especially when sperm are limited. This could decrease the chances of a male copulating in displays when multiple females are present. Females could also visit males in groups just because temporal patterns of mate search overlap. If that's the case, multi-female displays should be common, especially during periods of high nesting synchrony, as seen here in marine iguanas. From the male perspective, displaying when multiple observers are around may let preferred males attract additional mates, but could also create mating opportunities for subordinates who could sneak copulations while the dominant male is distracted by other females. Today, our goal is to demystify the context and reproductive implications of multi-female displays from the male and female perspectives in a lekking species, the lance-tailed manakin. Of course, this is a session on cooperation, and I'm talking about social context of courtship displays because courtship in this species happens in an unusually cooperative fashion. Males form long-term male-male alliances of dominant alpha and subordinate beta males to display for females, broadcasting duet songs, and then flying down to a low perch where they perform a two-male courtship display that ends with a stereotypical bounce and bow immediately before copulation. Male alliances are long-term, lasting up to 6 years. Male partners are not close relatives, and with rare exceptions, only alphas mate. Betas sire about 3% of all the chicks that we sample. Beta males do receive delayed direct benefits by increasing their later chances of becoming an alpha. I've monitored this study population since 1999, keeping track of individual survival, morphology, and age in a color-banded population, observing about 1,000 hours of behavioral observation with a research team every year to identify individual social status, and collecting blood samples from chicks to quantify reproductive success of males through genetic paternity. But the data I'll focus on today comes from video recording in a system pioneered by my co-author, Carla Vanderbilt. Carla developed a system of waterproof boxes placed about 4 meters away from male display perches. And in these boxes, we placed video cameras powered by 12-volt batteries that let us record for 9.5 hours a day at 12 display sites per year from mid-February until May in these 2 years. This gave us a great record of the timing and outcomes of everything that happened on these sticks during pretty much all daylight hours. And in 2013, we've scanned all of these videos to identify male and female identities from color bands that you can see on the videos. This approach worked. We recorded 1,295 displays for females, where a female watched males from the dance perch. And with our usual in-person observation, we used to see multi-female displays 1 or 2 times a year. So we thought it was a rare but exciting event. But here we found that 12% of displays had more than one female present, and that the rate of this occurrence was pretty consistent across years. 47 of the 95 uniquely banded females that showed up on display perches in 2013 actually were present for these multi-female displays. But there was no evidence that younger females were more likely to take part. So we found no relationship of female age category with whether they participated in multi-female displays. We found that the timing of multi-female displays varied by year. If females are using them to gain information, they should be more common earlier in the breeding season. But that was the case only in 2013, not in 2014. And even in 2013, the effect was quite weak. Maybe variable nesting effects, or maybe variable effects by year, could be because nesting synchrony varies among years. So to investigate that, we tested whether multi-female displays were more common when more nests were initiated in the 10 days after the display date. But we found the opposite pattern from what we expected. Multi-female displays were more common when fewer females were about to lay their eggs. What about outcomes? We found that multi-female displays were more likely to end with a copulation than displays to a single female. And this is in line with predictions of the mating synchrony hypothesis. So together, there is some support for this idea that synchronous searches result in multi-female displays, but that's not the only thing that's going on. And the reasons that these multi-female displays occur might be different for different females. From the male perspective, nearly all of the alpha males that we videoed observed or had these multi-female displays occur at their dance perches. Males that had more multi-female displays sired more chicks in the year, but they didn't get a greater proportion of multi-female visits. They got more single female visits as well. So multi-female displays weren't biased towards the sites of highly successful males. Interestingly, we found that beta males were more likely to attempt a copulation when two females were present. We defined a copulation attempt as when males performed a bounce or bow display beside a female, which is something that usually happens only in the final seconds before a copulation. So here you see a beta male doing a bounce while the alpha is slow flighting, but when he moves in for a copulation, the alpha attacks and drives him away. So most of these attempts were thwarted by alpha males, which is puzzling, as work in two other species of manakins with male-male alliances has suggested that females enforce a lack of conflict by not mating at sites where males engage in fights or where status cues are not strictly orderly. What's more, previous work suggests that alliances with cheaters are likely to split up. So did they? We asked whether these attempts decreased alpha chances to copulate. So after the display at hand, there was no difference in whether an alpha male eventually copulated in relation to whether his beta had attempted copulation or not. So alphas were unaffected for the display at hand. And in the bigger scheme of things, alphas with beta partners who tried to cheat actually sired more chicks that year. Alliances where the beta attempted to copulate were not more likely to break up. In fact, all 5 alliances where betas did not try to copulate did break up. So there's no obvious effect on alliance stability, though there was a lot of variation in how these alliances broke up. So in sum, multi-female displays occur regularly, but the reasons that they occur remain unclear, though we did find some support for predictions of the idea that search synchrony results in multi-female displays. Females seem to incur a risk of mating with non-preferred males when they attend displays with other females. From the male perspective, copulations in general were more likely during multi-female displays, and so were beta copulation attempts. Male-male alliances were not disrupted when these attempts occurred, and beta copulation attempts during multi-female displays provide a potentially important but context-specific route to beta reproductive success. For more information, check out work being presented tomorrow by Carla Vanderbilt and other work from members of my research lab, Meredith Kuzel, Pearl Rivers, and John Morton. Thank you for listening, and I'd be happy to take any questions.